Hello everyone, today we are going to discuss about the genetic code and its features. So, talking about the genetic code is defined as the relationship between the sequence of nucleotides in the DNA or its RNA transcript and the sequence of amino acid in a protein. Each amino acid is specified by one or more nucleotide triplets, which is also known as codons, in the DNA. During translation, the mechanism of translation will read detail in later. The mechanism of DNA replication, transcription and translation will read detail in later. But during translation, mRNA acts as a working copy of gene in which the codons for each amino acid in the protein have been transcribed from DNA to messenger RNA. tRNA, however, serves as the adapter molecule that couples the codon in the mRNA with the amino acid they each specify, which I will discuss later, thus aligning them in the appropriate sequence before peptide bond formation. Translation takes place on ribosome. Complexes of protein and rRNA that serves as the molecular machine, coordinating the interaction between mRNA, tRNA, the enzymes and the protein, factors requirement for the protein synthesis. Many proteins undergo post-translational modification as they prepare to assume their ultimate roles in the cell. So what we learned that the genetic code is defined as the relationship between the sequence of nucleotide in the DNA or its RNA transcript and the sequence of amino acid in a protein. Each amino acid is specified by one or more nucleotide triplets, which is known as codons in the DNA. That you should never forget. So now, talking about the features, the genetic code is unambiguous. That means each codon specifies only one amino acid. Only one amino acid. Each codon specifies only one amino acid, and each codon of 3 bases forms the triplet. For example, the U makes the phenylalanine. So U A makes the leucine. So there are 64 codons. There are 64 codons which are written in the 5' 3' direction. And the genetic code is degenerate reductant. That means most amino acids are coded by multiple codons. For example, most amino acids are recorded. For example, UUU, triple U codes for phenylalanine, and UUC also code for phenylalanine. So this both UUU and UUC code for phenylalanine. And the oval property. The codon that differed in the third, which is known as oval position, may code for same tRNA/amino acid. Specific base pairing is usually required only in the first nucleotide position of the mRNA codon. So another features of the genetic code is it is commaless and it is non-overlapping. That means genetic code is read from the fixed starting position as a continuous sequence of base. For example, U, A, like this. It is commaless and it is non-overlapping. This is just an example. And the genetic code is universal. That means genetic code is considered throughout the evolution. But there is some exception, that is mitochondria. It is the exception. So there is one start codon, which is the AUG, which codes for methionine, and there are 3 stop codon UAA, UGA and UAG. These are the stop codon. Thank you.